Hello, this is Mr. Hammond-Beyer again. We are still in the middle of a COVID-19 fiasco, so we'll be wearing masks in this class. But this is lecture number 2, basically a brief introduction to cells. It's a little more in-depth than APES needs, but it is just the beginning of what you need for anatomy. And so we're going to get into how COVID works. All right, a little bit of initial vocabulary especially for the anatomy folks. Anatomy is the study of the biological form of an organism. Physiology, the study of how the biological functions of an organism perform. So basically this is the structure, this is the chemistry, biochemistry of how this thing works. Okay, question. How does this jackrabbit keep from overheating? This is a desert jackrabbit. So it lives in the southwest United States where it's hot and dry, but it doesn't have very many sweat glands. You know, when we go outside and it's hot and we sweat, well, this doesn't have very many sweat glands. So what's its cooling mechanism? Notice the size of its ears, and so you have evaporative cooling in that particular case. Okay, and the capillaries are very close there, so when they get hot, the capillaries get a little closer to the surface of the skin, and that acts as a cooling mechanism. Okay, so form and function in animals is very, very closely related. And sometimes if animals have the same function, they get to that point, they end up having very, very similar forms. For example, let's look at these 3 examples. All right, a tuna, a penguin, and a seal. What's their function? Their function is to be able to swim through water with the least resistance possible so they can swim as fast as possible. Well, they evolutionarily came from 3 very different branches, but yet they ended up with the same or very similar types of forms. Okay? Very, very streamlined, aerodynamic so that they can pass through the water. That would be considered an example of convergent evolution, where very different organisms end up looking somewhat similar because of the environmental pressures that cause their form to follow the function. Okay? An animal's size and shape directly affect how it exchanges gases and energy with the surrounding environment. So 2 things that you ran into in earlier biology classes were diffusion and active transport. These are very, very important pieces. So think about an amoeba. An amoeba doesn't have a lung, they can't breathe, so how does it get oxygen into all of these parts of its cell? It has to diffuse or be actively transported across. Okay? Same thing with a simple organism like a hydra. Hydra only has a couple cell layers thick, so how does oxygen get in? It comes in this way. It ingests food, so its protein is digested here, and it has to diffuse out of this gastrocoel or stomach-like cavity to get to other parts. Now, think about and review the structures of a cell, because we need to understand this before we go on to the next piece. So take a second, pause the video, and go through and see if you can, in fact, label structures A through K. So on a piece of paper, on your notes, whatever, A through K, go through and see if you can figure out what all of these pieces and parts are. Okay, so as a review, here's an animal cell, here's a plant cell. The important parts are, here's the rough endoplasmic reticulum. That's the site of protein synthesis, which is very close to the nucleus and the nucleolus. Excuse me, nucleolus is concentrated messenger RNA. Nucleus is chromosomal DNA. And over here is smooth endoplasmic reticulum, the Golgi apparatus, which packages proteins for export. The centrioles, which aid in cell division. Whoops, sorry. Mitochondria, which store energy. And lysosomes, which are basically the stomachs of the cell. They help digest and break things down. Microfilaments help transport pieces around, give the cell some structure. But what I really want to focus on is the cell membrane. So if we look at, whoops, that's going the wrong way. How do we get to here? Okay, so let's look at the cell membrane. Cell membrane, okay, is a bilipid layer, if you remember this back from biology. So you have a phospholipid head and tails that are hydrophobic and hydrophilic, so that water, when you put water on your skin, it can't go directly into your skin because the hydrophobic part prevents it. But it is also embedded with a number of proteins. And attached to that, there are also some sugars, some glycoproteins they're called. And these are transport proteins. There's some cholesterol in here. Those molecules help give it some integrity. So what happens outside of a cell for, let's say that you are really thirsty and you drink a cup of water. And so the cells in your muscle, because you've just been playing basketball or dancing or whatever, the cells in here on this side need the water to help facilitate some of the metabolic things that are going on inside. There is diffusion from a high level of water across this cell membrane into the cell where there's a lower level of water. Okay? Active transport requires that these molecules that are a little bit larger, things like water and calcium and potassium, can diffuse across, but things that are bigger, like amino acids or even glucose, have to be picked up and pulled inside with active transport. That's why insulin is so important. It's got to attach to the glucose and then be actively transported across the cell membrane. Okay, so that brings us to how does coronavirus get inside of a cell? Because it's going to get inside the cells of wherever it is in your body, and there's got to be some, it's not diffusion because it's too big, so it's got to be active transport. Well, how is that active transport facilitated? There's a molecule called angiotensin converting enzyme, or ACE2. ACE2, it becomes important because it's ACE2. All right, so ACE2 is used by coronavirus to actually help facilitate it getting inside of the cell. So COVID-19 bonds to the ACE2 with what's called the S spike, or the spike protein, on its surface. And when this S protein comes up to the ACE2, it attaches to the ACE2. The S protein is cleaved into S1 and S2. S1 now has, once it's cleaved, S1 has the key that's going to unlock, basically, the RBD, the receptor binding domain, which allows it to get into the cell. All right. So S2 plays some role, I'm not exactly sure, in that membrane fusion. Okay, so let's look a little bit more closely at how this is going to work. So ACE2 is on many, many types of cells, and it's an enzyme that, in general, normally without COVID, is going to help break up some of these larger proteins, which are then going to regulate how fast and what kind of metabolism is happening inside of that cell. And where is it? Well, it's right out here at the surface, one of the surface proteins. Now, here comes along coronavirus. And if we look at a coronavirus, you know, in the lecture 1, we talked about that protein coat. So here's the protein coat, and attached to that protein coat, there are 3 important proteins sticking out. The first one, here's the RNA on the inside, and then let's look at it a little bit more closely. Here's the S protein, which can cleave when it comes into ACE2, into S1 and S2, which allows it to get in. There's also an M protein and an E protein, but we'll spend most of our energy talking about the S protein. Okay, so here comes coronavirus, and it's got the S protein up here, and it's going to run into an ACE2 molecule, which is then going to split and allow the virus to get in. Now, there is a thing called an anti-ACE2 antibody. And this, if that can get in the way, it can stop the S protein from attaching to the ACE2, which then is going to stop the virus from getting inside of your cell. Maybe you've heard about people who have had COVID, how they take their plasma and inject their plasma into people that are sick with COVID. And what they're looking for really is this anti-ACE2 antibody, so that it can get in there and block the virus from getting into new cells. Okay, so that's one possible treatment. Now, we know a little bit now, you know a little bit now about how COVID gets in with the ACE2, and I'm going to ask you to develop a hypothesis as to whether, to answer the question, is it important for COVID-19 infection based upon two things? Is it the type of cell that the COVID comes in contact with, or is it the location of the cells and the dosage? So in other words, is it a tissue type or a quantity of exposure that determines what type of, or how bad the infection is? Okay, so develop a hypothesis. Is it either tissue type or quantity? And whatever you say your hypothesis is, the null hypothesis is the opposite. So if you think tissue type, all right, your hypothesis is the tissue type is what's important with COVID infection, then my null hypothesis would be it's the quantity of COVID-19 exposure that determines the infection. All right. Now, if we change gears for a second and kind of talk about the types of systems in your body to help address that question, digestive, circulatory, respiratory, immune, lymphatic, excretory, endocrine, reproductive, nervous, integumentary, skeletal, and muscular systems are some of the systems of the body. We don't have time here to go through all of those, but we'll look at some of the tissues that make up those systems. So different tissues have different structures that are suited to their functions, okay? And there are basically four main categories of tissue: epithelial, connective, muscle, and nervous. Okay? Epithelial, connective, muscle, and nervous. We will talk about epithelial and connective today. We don't have a lot of time, so we won't talk about nervous and muscular at this time. Okay, now epithelial tissue is composed of three shapes. So there are three shapes: cuboidal, basically a square, three-dimensional square; a columnar, which is longer, taller than it is wide; and squamous, which is kind of flat. So think of squamous as a bunch of frisbees or floor tiles. They're kind of flat. Now, of those three types, then they can come in three patterns or arrangements. So you can have a simple layer, just one layer across, or you can have stratified, where you have a bunch of layers stacked up, or you can have pseudo-stratified, kind of partially stratified, of various lengths. So therefore you have three arrangements and three tissue types, or 9 permutations. There are 5 that are the most common. So let's look at those. So if we have a simple layer of cells, we have cuboidal epithelial cells. That's just one layer of cubes. Okay? And that would be, for example, in your kidneys would be an example of that. Now we can have simple columnar cells, which are long, thin cells, which are stacked, but there's only one layer of them. That allows things to diffuse out of your small intestine into your bloodstream, so that you can, the burger that you had for lunch, the protein that got broken down to amino acids, can get through here and get in. Okay? Another one would be pseudo-stratified, and these happen to be ciliated, and those tend to be in your nose and upper respiratory tract, because the cilia can now beat and push. Let's say you inhale some dust, it can beat and push that dust back out. In places that are going to get a little more movement of things, you have stratified squamous, so you have a bunch of them stacked on top of each other in case the top ones rub off when you swallow a big hunk of food that's not chewed enough. The top ones can rough off and it doesn't really hurt you too much. Okay? And then simple squamous epithelial are, for example, in your lungs, where you have to absorb oxygen and release carbon dioxide very quickly. You don't want to have a real thick cell like this one because it would take too long. Okay, and here's an example of some columnar epithelial cells, and underneath it you've got some connective tissue. Okay, so connective tissue is basically tissue usually that binds things together. Okay, so it contains sparsely packed cells, and they can be throughout any kind of matrix, and it all depends. And there are 3 types of connective tissue: collagenous, elastic, and reticular fibers. Okay? Collagenous, think of collagen. Your hair has some collagen in it. Elastic fibers are things that can stretch. And then reticular join your, for example, especially reticular fibers, join the tissue to adjacent structures. Okay? Now, they're also included in here are fibroblasts and macrophages. And macrophages I need to talk about because of COVID, in that these form B and T cells, and those are part of the immune system, and they help attack COVID-19. So if a person is exposed to COVID-19, they will develop B cells and T cells to attack the virus and have some memory of that to give them longer-term immunity. Okay, then there's loose connective, cartilage, and fibrous connective tissue. Those are not too important in this particular example. Adipose, blood, and bone, and those haven't been indicted with COVID too much. But here are some examples of these types of tissue. So the loose connective tissue would be the tendons, and here's a good example of a tendon. If you grab right here, you can feel that tendon. Here's the muscle. In this case, it would be the quadricep. All right, and then fibrous connective tissue gets down a little bit deeper, is going to attach to the bones. Adipose tissue is fat. Cartilage is tissue that's going to protect the bones. The bone tissue itself, blood is a connective tissue, and the nuclei of the muscles. Okay, so here we are. We'll spend a lot more time later in anatomy on this, but here's my parting question. So where does COVID-19 enter the body, and which tissue is most susceptible to COVID-19? Now go back to our early discussion about ACE2 and do some research which of these types of cells and tissues have the most ACE2 receptors might help you answer your original hypothesis. And that concludes lecture 2.